everyone. Welcome to Jumpindetox. And tonight we have a special guest for our show. It's someone that I really appreciate since 2012. And he was also my professor in Geneva when I was studying nutrition and detoxifications. And since these times, we are a close friend. And I'm very happy to have on my show Boyd Ali. So thank you, Professor Boydale, to jump in my show, and I'm very excited to have an interview with you because I know you work since many years, but for my followers, I really want them also to know you better. And so, how are you today?
I'm doing good.
You're doing well. Okay, great. So you are based in USA, and I remember that in 2012 you were in Geneva sometimes to make introduction about mercury intoxication. And for me, it was the starting point. And in 2013, I organized also a very interesting conference for you in the medical faculty, and it was very, very great. And for my first question for you will be, what was the starting point with mercury intoxication? When was the first time you just heard about that?
Well, I had a graduate student that was working in the Alzheimer's Research Center at the University of Kentucky, and she came to me and said, she came and revisited the lab as a postdoc, and she told me that there was no idea what caused Alzheimer's disease, and I had developed a technology, and that's what everybody should understand. I was funded 25 straight years by NIH for this technology I've invented called nucleotide photoaffinity labeling. Too complex to talk to on a show like this. Did that. We decided to test and see if we could see the changes in normal brain versus Alzheimer's disease brain. And we did that, and we found major biochemical abnormalities in Alzheimer's disease brain using this technology that was funded by NIH for 25 years. And we published that, and everybody said I was right. But then, whenever you something that causes a brain abnormality, you try to find out what it is. In other words, what could I add to normal that might mimic the same biochemistry we saw in Alzheimer's disease brain? And at that time, people were thinking it was aluminum. And so I tested all the toxic heavy metals, and only one toxic heavy metal mimicked the abnormal biochemistry or induced the abnormal biochemistry that we saw in Alzheimer's disease brain, and that was mercury. And I published that, and that's when NIH didn't want much to do with me anymore. They didn't want to hear that mercury was probably the cause, or at least an exacerbating factor in Alzheimer's disease. And I think
Yeah.
the reason for that is that the FDA and the National Institutes of Dental Research had all supported the use of dental amalgams in American mouths, and that is the major source of mercury in Americans, and I think Europeans also, is the
Hmm.
number of dental amalgams they have.
Yes.
And
Yes.
we showed also that you could take a tube of distilled water and add to that tube an amalgam that was been extracted from a person's mouth in a tooth, and let it sit there for just a few minutes, and take that aliquots of that water and add it to the normal brain homogeneous, and induce the same abnormality that you see in Alzheimer's disease. So that's a fairly direct correlation. And it has not been very popular. It's made me very unpopular, I think, with the FDA and with the American Dental Association, and maybe even the American Medical Association. Uh, that very likely it's a toxicity that's causing Alzheimer's disease.
Hmm. Hmm. Okay. And we are going to see a short video, and for me, it's quite important for the people to understand that. So let me just doing like that. So this video named "Smoking Tooth," and before talking about how mercury can harm the brain, because mercury harm everything in the body, but also for the brain. And you just talk about Alzheimer's disease, and also perhaps we are going to talk about Parkinson. Let's have a look about this video, this short video. Huh?
Silver amalgam fillings constantly leak mercury. This lighting allows us to see mercury vapor from a 25-year-old silver amalgam filling. This filling was rubbed with a pencil eraser for just a few seconds. It will
This is
continue to
vapor.
outgas for over an hour. This amount of mercury is 1,000 times higher than the EPA allows for the air we breathe. View our website at holisticdentistpgh.com to learn about the serious health risks of having mercury in your body.
So this is quite amazing to see the vapors coming out those amalgams.
Yeah. And that's what people have to understand. That vapor is not very reactive. I mean, it comes off and it gets in your body, and that's the reason it's so dangerous. When it gets in your body, it likes getting into the lipid bilayers or into the hydrophobic aspects of the body, and it doesn't react. It doesn't react with the protective proteins that might be in your blood,
Hmm.
etc. What it does is it moves around the body and it collects in the central nervous system. When it gets into the central nervous system, there's an enzyme there that oxidizes the mercury from Hg0, the vapor, to
Hmm.
Hg2 plus, which is the most toxic form of mercury, and it gets trapped in the brain. And the trapping of the mercury in the brain, then it starts reacting with sulfurs. And in the brain, there's the electron transport system in the mitochondria where they have iron sulfur centers. So mercury displaces that iron, and you get a buildup of unbound iron in the central nervous system. And the mercury inhibits the electron transport system, prevents you from making ATP and causes problems. But it's all something you can bypass by having just a lot more mitochondria. But the action of the unbound iron causes an oxidative stress in the
Hmm.
Alzheimer's brain, which mercury can't do by itself. Mercury is not a redox metal; it cannot donate electrons and produce hydroxy free radicals. But the iron it displaces can, and it does, and it builds up. But the iron is trapped in the central nervous system. In other words, it can't be excreted very effectively. It stays in the brain for a long time, generating oxidative stress, which is the major chemical damage done to the Alzheimer's, and not only the Alzheimer's brain, but the Parkinson's brain, the Huntington's disease brain. Uh, it is the major one of the major toxicities done by mercury. Mercury has its major toxic effect, in my opinion, on the human body by displacing iron, causing unbound iron buildup in the body,
Hmm.
especially in the central nervous system.
Yeah, yeah. We're just showing to the people that there's also a study that you participate to on January 2022, talking about this genetic brain iron accumulations. And this is very interesting because you are also the CEO of a laboratory creating a new kind of chelator able to detox heavy metal like mercury, arsenic, or lead, but also iron. Can you talk a little bit about that?
Yes, if when we get to see the structure of NBMI, we see that NBMI has two long arms that are very similar to the end arms on glutathione, and those
Hmm.
iron those two sulfurs can move around in different positions in the body or in the
Hmm.
space, and it binds all the toxic metals. If they bind sulfur, a single sulfur will come out from NBMI, bind to the metal, and then hold it there, and then the next sulfur will come around and bind it on the other side at a different position. So it has two sulfurs that bind each toxic metal. In addition, it has two nitrogens with unshared electron pairs that can be involved in the binding also. And if you look at the binding and doing the testing,
Hmm.
NBMI binds every toxic metal that we've ever tested with it, including arsenic, mercury, lead,
Hmm.
cadmium, uh, and uranium.
Hmm.
So it's what I call an induced fit. And what this is that the two arms, and I'm going to use my hands here a little bit. The two hands, if you look at they can move around, And as they move around, they can orient to the coordination chemistry to fit that specific metal. Because iron and mercury and cadmium do not have the same coordination chemistry. And that's a little complex for people to understand. But you have to be able to change those sulfurs if you want to get maximum binding. And that's what we have. And so we have and everybody should understand the simple take home lesson is that NBMI, when you take that and it gets in your body, it not only binds mercury, it binds cadmium, gadolinium, it binds a large number of even lead. It will bind it very, very tightly because it can have an induced fit for the specific metal that it first runs into. And so
Yeah.
so when you take NBMI, you're detoxifying several toxic metals at one time. You can't do one and not do. You can't do mercury and not do iron. Let's put it that way. I mean, you have to. They both get done at the same time, roughly the same time.
Yeah. For the people who are looking, and NBMI, it's the chelator that you created. Um, Chelation therapy has a long story behind, and chelation therapy helped a lot of people in the past, and today they have a lot of very good results. But we need to be very careful about these techniques. Can you talk a little bit about how we can be very we need to take time and to think about how we can really detoxify the body.
Yeah. Well, the main difference between NBMI and other chelators, and the way the general medical community would look at it, they would say you detoxify the metals by getting them to be excreted from your body.
Mhm.
And with NBMI, that's not true. NBMI, I mean, like if you look at the paper we have with Clark et al., where we gave rats 1 and 2 and 14 times a lethal dose of mercury and then treated them with NBMI and totally prevented the 1 and 2 lethal doses from killing any of the rats, but the mercury was still in the body. And so, unlike EDTA and DMSA, which bind mercury and carry it out through the urine, NBMI binds it into an inert complex while it's still in the body and eliminates the toxicity within a few hours. And then it gets carried out very slowly by the P450 system of the body that detoxes every hydrophobic molecule that gets in there to get rid of it. So the compound that works totally different than other chelators. In other words, if you look at the study where we did the effect on H670 rats, where we bound the iron in the brain and stopped all the oxidative stress, yet some of the iron was still in the brain. And so it does that because NBMI, because of its design with the 2 sulfurs and the 2 nitrogens in those arms, totally cover the electron shells of the mercury and the iron and other metals, eliminating their toxicity. They become inert. They become inert objects in the body that are excreted slowly, but they're totally without toxicity. So,
Yes.
and see,
Yeah.
it does decrease the iron, but it decreases the oxidative stress much faster than it gets rid of the iron.
Yeah, completely. And I want also to show something quite interesting. Also, it's what is the effect of mercury on neurons? Because your chelator is able to detox the brain because it's able to pass through this brain barrier. And I hope that you can give us some more information why he's able to do that and why your chelator is quite revolutionary about that. So let's have a look about this video. It's just an amazing video.
Shown here is the neurite of a live neuron isolated from snail brain tissue, displaying linear growth due to growth cone activity. It is important to note that growth cones in all animal species, ranging from snails to humans, have identical structural and behavioral characteristics and use proteins of virtually identical composition. In this experiment, neurons also isolated from snail brain tissue were grown in culture for several days, after which very low concentrations of mercury were added to the culture medium for 20 minutes. Over the next 30 minutes, the neurite membrane underwent rapid degeneration, leaving behind the denuded neurofibrils seen here. In contrast, other heavy metals added at this same concentration, such as aluminum, lead, cadmium, and manganese, did not produce this effect. To understand how mercury causes this degeneration, let us return to our illustration. As mentioned before, tubulin proteins link together during normal cell growth to form the microtubules which support the neurite structure. When mercury ions are introduced into the culture medium, they infiltrate the cell and bind themselves to newly synthesized tubulin molecules. More specifically, the mercury ions attach themselves to the binding site reserved for guanosine triphosphate, or GTP, on the beta subunit of the affected tubulin molecules. Since bound GTP normally provides the energy which allows tubulin molecules to attach to one another, mercury ions bound to these sites prevent tubulin proteins from linking together. Consequently, the neurite's microtubules begin to disassemble into free tubulin molecules, leaving the neurite stripped of its supporting structure. Ultimately,
So I guess, why we don't talk about that, because we think that Parkinson or Alzheimer disease, just came from nowhere and that it's a generations disease. But we have answers about how mercury, because all those people with mercury in their mouth, also on the fish like tuna and salmon, we have a worldwide mercury intoxications going on. So what's going on?
Well, I think our government is not very honest about. They want something. There's a huge market out there. If we could develop a therapeutic treatment for Alzheimer's disease, you would make billions of dollars.
Yeah.
They don't want to prevent it. They want it to. They don't want to look at it and say we know the cause. Let's eliminate the cause, and
Mhm.
people won't have Alzheimer's. The drug companies don't make any money when you do that. That's my best answer because I can't understand why they don't jump on this.
Yeah.
The science is very solid. I would tell you that uh, video you just showed is not exactly right. I didn't make that,
Yeah.
but some people did it. And what it is, NBMI and mercury does not cause the tubulin to not aggregate. It causes it to aggregate improperly. And you can see that if you take, and I did this and I published it, if you take control brain and take it to 0 degrees and spin it down, most of the tubulin will be in the soluble aspect of the solution. It won't go down in the pellet because it's all in monomers and dimers. If you treat that same normal tissue with a little bit of mercury, all the mercury will go down in the pellet. In other words, it abnormally aggregates. It doesn't float away free like they said in that paper. It actually abnormally aggregates. And if you take an Alzheimer's disease brain and homogenize it and spin it down, all the tubulin will go down in the pellet. There won't be any soluble tubulin like you normally find in normal brain. It doesn't do that. And so the mercury is causing the tubulin to abnormally aggregate and come off of the neurons and producing the neurofibrillary tangles. That's been published by me a long time ago. And it fits into what causes Alzheimer's disease and other neurological issues. Mercury get in there, depolymerizes, causes an abnormal polymerization of the tubulin, and it cannot bind to the neurofibrils. And that's the reason you get neurofibrillary tangles formed in Alzheimer's disease.
Yeah. Yeah. Can you talk a little bit also about your company? Because the chelator you created a few years ago was used many years in US as a complementary dietary, and now you are still in the process to have FDA approval as a drug. What happened?
Well, this compound, let's express it properly. NBMI, we call it NBMI because that's easier to say than the whole name. But the compound is not only a good chelator, it is one of the best antioxidants ever made. In other words, it scavenges hydroxy free radicals in a test tube better than anything ever made that you could possibly swallow. And so we sold it 2 years uh, to as an antioxidant. And I thought that was important at that time because if you consider autistic children, who are, there was a big increase in autism in the United States in the late 1980s. And and you can argue about what did it. I thought it was mercury, but they also, one thing everybody agrees on is that they suffer from oxidative stress. Autistic children suffer routinely from oxidative stress. And our compound was an antioxidant, and its problem, its chemical process for stopping oxidative stress is that it scavenges hydroxy radicals that are inexistent, but it also binds the iron and the mercury that induce the production of those hydroxy free radicals. And we sold it, and because it was non-toxic, and it's made out of two natural products. If you look at the structure, it's made out of benzoate coupled to two cysteamines. And those are both natural products, and the benzoate is found in beer and everything as a preservative.
Okay.
And so when we started selling it, we went very carefully. We only gave it to medical doctors and to dentists to treat their patients. And we said to watch it, and we did a lot of safety studies. We sent a form to the FDA showing that you could take rats and give them 1,000 milligrams per kilogram body weight, which is a huge amount, for 28 days and not make them even sick.
Wow.
And we did that with several hundred rats, and we showed that to them. And so the compound is just not toxic. And and you're looking at somebody, I've been taking the compound myself personally for 16 years, and I'm 82 years old now, and I don't have any quivers or tremors, and my brain is the thing that works best on me. My legs don't work so well, but my brain works real well. But anyway, when we did that, what happened was that the mothers on blog sites, when they started getting it out, and I gave it to doctors first that were treating autistic children, and they had very good results. And then so more and more mothers got into it, and the mothers, they're mothers. I mean, they're going to try and protect children. They're going to try and help other mothers. And so on the blog sites, they started telling everybody that NBMI was the best best treatment they ever had for their autistic children. And especially it stops the intestinal dysbiosis that autistic children have. And we have tested that with the highest researchers at Ohio State University Medical Center. And NBMI prevents mercury and thimerosal and other drugs from causing gut dysbiosis or leaky gut syndrome in a laboratory test. I mean, the compound has everything going for it. I mean, if you sit and talk to researchers, if they wanted to talk to you, you would say the reason it stopped the intestinal dysbiosis and prevented leaky gut syndrome, because certain toxins cause the intestinal membranes to become very leaky. And NBMI prevents that. If you add that to the test system and add the mercury, thimerosal, it totally prevents it because it binds it and renders those compounds non-toxic. But that's the reason the FDA shut me down, because somebody in the Pediatrics Association complained to them that I was using a hardcore drug to treat autism and I was going to kill a bunch of children. Well, the bottom line is we have now taken NBMI through every test that the FDA would require to show that it's not toxic. The compound is not toxic for a drug. It's less toxic than aspirin, much less toxic than aspirin. And yet we're having a hard time getting the FDA to approve it. They say we now agree that your compound is totally safe for all the animals we've tested and for humans. And then they say it's efficacious for rats because we can take rats and give them multiple lethal doses of mercury and save them by following it up with NBMI, either orally or by injection. And they say, and your compound is very efficacious at preventing death from mercury in test animals. But we don't know that that's the same in humans. You know how hard that would be to do in humans? The first group told us we had to do it through the animal rule. You can't do that kind of testing on humans. Give them a toxic metal or find people that are mercury toxic and do it. And so they told us we had to do it under the animal rule. So we developed a very refined and extensive study where we induced kidney toxicity in rats by injecting mercury under the flap of their skin on the stomach. And we could do that and induce it, just not kill them, but induce measurable toxic effects on the kidneys, which we proved by histology and by looking at kidney toxicity biomarkers. And then when we gave NBMI, the thing we found, to be very short, NBMI given ahead of time would prevent the mercury toxicity, And if you induced the mercury toxicity and then gave NBMI, it would reverse it because the kidney can repair itself pretty well. And we thought that would get the FDA to say, yes, do this test, do more studies on the same animals, and we'll approve this. But they didn't. They said, no, you have to do a human efficacy study. And when we asked them, well, where do we find these mercury toxic people in the United States? They couldn't tell us because they don't think that people that have 15 amalgam fillings are mercury toxic.
Wow.
And so, I mean, it's a conundrum that it requires somebody that really wants to solve the problem. And I don't think our FDA really wants to solve this problem.
Yeah.
I mean, that's the opinion I get by talking to them. They never call me. They never, for example, when they shut me down, it's because they said I was using a compound that could be used to treat a medical problem called autism, and therefore, it's not a supplement anymore. It's now a drug because it cures a disease or affects a disease very dramatically, as a matter of fact. I mean, some of the autistic children that took NBMI totally came out of autism diagnosis at that time. Yeah, I know there are a lot of their stories are all over the place, but I have yet to have anybody from the FDA or the American Medical Association call me and say, "Why? Let's discuss your compound. Why does it work, and why is it not toxic?" They don't want to learn this because there's no money to be made. What I'm doing is I'm damaging the income of the pharmaceutical industry and the medical industry by having something that can treat what is the most common probably affliction in the world, and that's eating toxic food. And right now, there's a big fight going on that they're saying that the FDA, the Food and Drug Administration, that they are not monitoring the food in the United States carefully enough. And not only babies, but adults and everyone else are eating foods that are very toxic, especially with heavy metals like mercury and lead. Lead's in our water everywhere. And so there is not a sincere effort, in my opinion, and I'm saying it up publicly, by our government to try to render treatment for people that are heavy metal toxic. They just say, well, we don't see it happening. Well, you got to be blind and stupid to not see it happen, because the NHANES study, the National Health and Nutritional Evaluation Survey, I think it's done by the EPA, says that the mercury levels have gone up in Americans dramatically in the last 10 years, even much higher than it was before. Wow. And that's, everybody can go to NHANES and look at it, and you see that you have mercury levels that increase in people, especially after they turn past 18. The level of mercury in the blood of older people is dramatically high, and they're the ones that get affected the most.
Chelation therapy is a huge market in US, and I think perhaps in some countries in Europe, because it's not in some countries it's not allowed, like in France or other countries. But chelation therapy was also is also used concerning the heart diseases. I think cardiovascular operations. They use EDTA, so it's a kind of chelator. But this market, those people who work on this old generation of chelator, are they participating on that? Are they fighting you in a way that, because they are going to see your chelator, your new chelator has something that's not very good for the business?
Well, I think that's a possibility. I don't know. I mean, they don't write me, and they don't challenge me to come and give a debate on a seminar. But calcium EDTA by itself is very toxic. It'll kill humans, and it kills them by chelating essential metals. And the same thing with DMS and DMPS. They both remove essential metals from the body, and our compound doesn't. And so the reason that the chelation has been outlawed in a lot of countries is that it induces more medical problems than it absolves, and that's because they're using the wrong type of chelator. I mean, they're using a chelator that chelates zinc and copper and takes that out, so you have a decrease in essential metals. And so they have a reason for not having those chelators. But what they need to do is to make a new set of chelators. And that's the reason I invented or developed NBMI, is
Mm-hmm.
because I could see, and we've studied a lot of it. The compound is really very, very good, and it
Yeah.
does treat oxidative stress. I mean, people. We treated 10 patients, two doctors that had an autistic child each
Mm-hmm.
worked with us on something that we published, and we gave them NBMI, and they gave it to 5 autistic children and 5 older people. They didn't have any adverse effects come up at that time, and what they found was that we measured over 200 items, to see if the compound was having any effect on any metabolism or production of any biological response modifiers, etc. And the major issue, we didn't see anything that was of any concern. But what we did see, what the researcher did see, who was an expert from NIH, by the way, on measuring glutathione levels, was that the glutathione levels in the people who took NBMI or OSR number one at that time, the glutathione levels went up dramatically. And I had my glutathione levels checked when I was 65. That was some years ago. And I had the glutathione levels of a teenager because I'm taking the OSR.
Okay.
It stops the oxidative stress. And see, that's how you get rid of, when you age, your glutathione levels drop because the oxidative stress levels in your body goes up.
Yeah, yeah,
And
yeah,
so older people don't have as much glutathione as younger people. But if you give them an NBMI, their glutathione levels go back to normal.
yeah.
I mean, like they're
Let's talk about glutathione because on the detox field, that's why I wanted also to debunk what is detox, because we are talking sometimes about well-being detox, drinking juice, take some spirulina and all this stuff, and we have also the real detox, with your product and the way how we can treat people with teeth to make a safe removal. So there is a lot of ways to do that. But today in those kind of clinic, sometimes we inject glutathione directly. We have a kind of glutathione business on capsule. So, and I remember one time you just say that this glutathione is not efficient because it needs to be created inside the cell. So let's talk about that a little bit, about this glutathione business.
I'm not sure I understand exactly what you're getting to, uh, but what you
If I
want to do?
take if I take glutathione, by
Oh yeah,
on capsules or by intravenous therapy, they say okay, it's a antioxidant treatment, etc. And I remember you saying that glutathione, the best one is the one who is created inside the cell.
yes, because glutathione is made, evolved, however you want to say it. But glutathione is uh, 3 to 5 millimolar inside the cell, and it's
Hmm.
1,000 times less high. Lower in the blood. And why is that? That's because when glutathione gets into your blood, it goes to the liver, it goes through the biliary transport system, and there are receptors in there that bind glutathione and put it in the bile and whatever is attached to glutathione. That's how you use glutathione to get rid of toxic materials.
Yes.
And so when you take glutathione orally, it gets into your blood before it gets into your cells. And so that glutathione is probably going to be picked up by the biliary transport system and put into the bile and into the feces. It's not going to get inside the cells. No one has ever published a paper showing that glutathione that you take outside the cell gets inside the cell. It just doesn't. And especially if it's going through the plasma first and going through the liver, because the liver is going to clear all the glutathione out. And if you look at the P450 system, which is the major detox system of the body, and if you have hydrophobic compounds, like benzene, etc., uh, or drugs, what happens when they get in the body? They go into the fatty tissue, and then you have the P450 system, and you have
Yeah.
all these oxidizing enzymes, CYP enzymes, they call them
Hmm.
CYP, and they oxidize those hydrophobic compounds because you have to oxidize them in the first step to make them soluble so you can get rid of them. And so
Yeah.
they'll put a
Reduction.
hydroxyl group. And in our case of our compound, if you have mercury NBMI, you put a hydroxyl group on that aromatic ring on the NBMI, and that doesn't get rid of it. But we've shown that we can show that oxidation step occurs, but with the CYP enzymes or with 2 or 3 maybe, and then There is a phase two of the P450 system. You attach something like a sulfate or a glutathione molecule to the oxidized hydrophobic compound, and that attaches a charge to the compound and it makes it water soluble. In the case of NBMI, you attach glutathione because it goes out through the feces through the biliary transport system. As it gets soluble, that glutathione is carried, glutathione modified NBMI mercury complex is carried out into the plasma, goes through the liver, the biliary transport system grabs it and puts it in the feces. And if you take rats and you have a mercury toxic, you can look at the level of mercury in their urine because they're getting some of it out that way. But when you give them NBMI, it shifts and most of it goes out through the fecal route. You don't get hardly any out the urine when it's a mercury NBMI complex. And so it's working like it should work if the people understood the system. But it's as a scientist, I can tell you I'm very proud of how it worked out. And I didn't engineer that into the compound because I'm so smart and predictive. I did follow your nose research. If this compound works, it doesn't make you sick. Well, it doesn't interfere with any metabolism at all. Only thing it does is it scavenges hydroxy-free radicals and binds toxic metals. It doesn't go and inhibit anything. It doesn't pick up essential metals and take them out of the body. We have done this extensively. When we did the rat study, we showed that even though we were giving 1,000 milligrams per kilogram body weight, which is a huge excess for treating that you would need to treat anything, we didn't lower their iron or zinc levels at all, or copper levels. So the compound is incredibly safe, and it has marvelous properties that makes it something that should be available to everybody in the world, especially people that are in gold mining areas where they're using mercury to extract gold. It would save their lives.
Yeah.
And here's the problem. I think our FDA is weak in science. When they looked at the Clark study, and then everybody here can get a copy of that Clark study if they want, where we showed that if you injected rats with multiple levels of lethal doses of mercury, and then followed that 20 minutes later with an injection under the flap of the front skin, stomach skin, you could save their lives. We saved 100% of those that got 1 and 2 times the lethal dose. Saved 75% or about 67% of those that got 14 times the lethal dose. Nothing else in the world can do that. I don't know of anything that can do that. And yet the FDA said this was weak proof of efficacy. In a report to us, I got a copy of it. They said that's weak proof of efficacy. How do you argue with people that just seem to not know any science at all? That is the best proof. And I know because of reviewers of my paper, when I submitted it for publication, came back and said, my God, this is phenomenal. And the FDA looked at it, well, it's weak. I mean, they don't want to approve it. I get the very distinct impression that the FDA does not want to stop this because I think there are a lot of good people at the FDA. I want to make that clear. But you have politicians appointed at the top and are told, you make sure that our pharmaceutical industry gets served first. And those people, they don't want to lose. They don't want people to be totally healthy. If they're healthy, the pharmaceutical industry, just think what it would do to it. If you could eliminate all the neurological problems and the intestinal problems like leaky gut by taking a compound that just protected you from toxic metals that you're exposed to, according to another government agency, says Americans and especially babies are getting exposed to a large number of toxic metals. But you got to have somebody in power that cares about human health and not about profits for pharmaceutical industries. And I don't think we have that in America anyway. And this spreads out because if you don't get it approved in the United States, you're not going to get it approved any place in Africa. I mean, they're just going to say, well, if the Americans won't take it, we're not going to take it.
Yeah.
And they need to wise up. The United States is not really concerned. The United States regulatory agencies are not really concerned about the health of Africans or South Americans. I mean, they're not. And that's the reason we did a study on the Ecuadorian miners. And, when we did that, giving 300 milligrams a day, which is too low, but that was a study, that's what they would allow us to do at that time. We saw a drop in the urinary mercury level, even though they were breathing high levels of mercury every day. We saw
Wow.
it drop in 10 of 11 people. And so the compound worked in spite of being exposed to that mercury every day. And so uh, I think that this compound needs to be uh, it needs to be approved worldwide. I mean, there
Yeah,
needs
yeah.
to be somebody look at it. And I'm getting too old, Nicholas. I mean, I've been fighting this now for 20 some years, and the drug is there. It's been tested. It's available. Anybody that wants it can possibly get it. And the Chinese chemical companies are uh, doing black market on our compound and
Mhm.
getting it around the world. And so other companies are doing that too. And I really can't blame them because they need it.
Mhm.
And what will happen in the United States? We'll be buying NBMI approved as a drug in China. It'll never get approved in the United States.
Yeah, because your product is efficient for COPD, for obstructive pulmonary disease, huh? I think.
Yes. Well, look, anyone that has an illness they're interested in, just go to Google, type in the name of your illness and oxidative stress. Oxidative stress is associated with almost all it's all toxins. Just about all toxins induce toxicity by inducing oxidative stress. And how they do
Mhm.
that is they knock iron off of iron binding proteins. And when the iron is displaced, it becomes very toxic.
Yeah.
And so, I mean, uh, it's tough to get this even discussed. I mean, because when I reported that mercury caused the same biochemical abnormalities you saw in Alzheimer's disease brain, you would think I would be invited by the Alzheimer's Association or the NIH to symposium and conferences on Alzheimer's disease and possible treatment. They shut me down. They took away all my grant money. I mean, after being funded for about 25 straight years, the NIH wouldn't fund me anymore. They wouldn't
Mhm.
even. It got to the point where they wouldn't even consider my grants because they said we're just not interested.
Mm.
I mean,
Shame.
it's not been easy on me. I can tell you that.
Mhm.
And so I would just tell people that are interested, try to get it approved in countries where the FDA cannot prevent you from getting it approved,
Exactly.
and try it. But it's sort of like ivermectin and COVID. Ivermectin worked really well in India at preventing COVID infections, and yet
Yeah.
the United States approach, because it was out of a patent, is that ivermectin didn't work and we shouldn't even be using it. I think we're facing the same situation with NBMI. If we stop all the heavy metal toxicity by people taking uh, something like this that are in a toxic situation, which you can measure in the urine, uh, we're going to cost pharmaceutical industries a lot of money.
Mhm. Mhm. We are saying today, and WHO also say that a high percentage of health issues are directly linked to heavy metal intoxications. And what will be your message? The thing that you want to say to the next generations of scientists, doctors, searchers, researchers, to be more involved on this topic about heavy metal, because I'm quite young in this topic since perhaps more than 10 years. I'm not a doctor, I'm a nutritionist and in detox, but I'm just amazed to see how there is not a few people like you. There is just few people, and very with a strong mind and with a lot of things to offer. But what could be your message for the next generation of scientists?
Well, my major weakness is I'm not very politically active. I mean, I'm not very. I never have been. I've been a scientist, and right now the main blockage to getting good medicine is politics. And the fact is, I think our regulatory agencies, not just in the United States, in Europe also, and other countries, has been compromised and taken over by uh, people from the pharmaceutical industry who want to use illness as a profit making process. And so we have to get the politics out of treating medical uh, uh, issues like this, because this is And I think my compound is a perfect example. Ivermectin is too, so is hydroxychloroquine. These are compounds that were invented and treated, and then they came out of patent protection, and then all of a sudden they no longer work. And why does that happen? Why was ivermectin so popular, and then all of a sudden it's not popular, and it can't be used, and it's considered something that doesn't work? And we had major people in the medical industry in the United States saying ivermectin doesn't work. And I haven't done any of the research, so I don't know. But when I hear that a country or a state in India lost most of their COVID infections by giving ivermectin, I'd say that that's something we ought to look at least. And in this country, they say no, just like they did me. We don't want to look at that. Mm-hmm. We don't want to look at that. We might see a mistake we made, and we might find something that would prevent a disease that's making a lot of money for the medical association.
Right. Same.
Yeah. So let's I mean, really, the main thing that young people have to do, and if you look at medical doctors, like there were several that were coming up with using ivermectin and other ways of treating COVID, and they lost their license. I mean, people went after them, and so how is this? I mean, there's just major politics that need to be corrected in the organized world if we're going to get rid of some of the things that are very toxic and going on.
Yeah, and for sure that I think people now will know you better and about also your compound. We met in 2019 in a Met Detox conference in Berlin. This kind of events are going to be more and more in the future, you think?
I hope so. I mean, that's what's needed, but they got to be organized by people that want to see change, not by people that just want to come up and protect their business. I mean, if they're going to have chelation conferences with chelators that don't even enter cells, and that's a major thing they push. I mean, that is what is just boggles my mind that people say these chelators are good at detoxifying people, and they don't even get inside a cell. And that's where the toxicity is occurring. It's not occurring in the bloodstream; it's occurring inside the cells, inside the neurons, and they can't get there. And so they say, but our compound does a great job because we does a good job making them money, but it doesn't help the patients very much.
Yeah. I'm quite, and I think I was really amazed when I see all your papers that your compound pass blood-brain barrier and the cell barrier. So because this is quite unique, and yeah, I hope that people will one day have the possibility to use your compound, and I know that we are all working on that, and we are going to help this day coming one day,
Yeah.
So
yeah. Well, that was the one concern that the FDA had that they held on. So they said, does your compound build up in tissues and not get excreted? And so we made radioactive NBMI that you could follow. And we spent several $100,000 getting this synthesized and tested in rats that were some of them were, half of them were given mercury so that we knew they were mercury toxic. And we gave the drug, and we could follow the radioactivity throughout the body of the rat by autoradiography or by taking the tissues. And NBMI goes into through the gut. We can follow as you absorb it. It goes in the stomach, then it gets in the blood, and then it gets in all the tissues of the body. And it peaks at about 2 to 4 hours in the plasma. And it goes from the plasma into the cells, including the central nervous system. And it stays there for a while, and then it starts going out. And in 72 hours, it's all out. I mean, I'd say 98% of it is out of the body, whether it bound mercury or not, because it's going out through the P450 system. I mean, the detox system that the body is well defined in the human body. And so this compound does not build up in your tissues. It doesn't cause any eye problems. It didn't cause any headaches. It doesn't do anything. It might cause people that are sulfur sensitive to have a rash, That's the worst thing we've ever heard of it. Is it might, and that's those same people get the rash if they take glutathione, which is a natural compound. So the compound is totally safe. It's very effective, and it needs to be put out because I'm a basic research scientist, biochemist type. I cannot treat patients just like you can't. I mean, I shouldn't be allowed to, and I
Mm-hmm.
agree with that.
Mm-hmm.
But so if the FDA can keep it in my hands, they can keep me from doing, unless I get millions of dollars of donations, any studies on humans. But if you put it out and make it available for the regular medical doctor to treat patients that walks into his office, you're going to find out that this compound can solve a lot of medical problems. And of course, that's going to cost the medical association and pharmaceutical companies a lot of money.
Mm-hmm.
And so it's going to be a political fight if it ever gets through. And I have severe doubts that in my life that I'll live long enough to see it, because there's nothing rational about what the FDA is doing. It's not
Mm-hmm.
rational. It's not good science. It's not good medicine. It's not good for the American people. And basically, the only people it's good for are those who want to make drugs to make money. And
Yeah.
that is the basis of American medicine: is make a drug that's patented that you can make money. If it's not patented anymore, like ivermectin, don't sell it, because the money it may decrease the amount of medical treatments needed, but it just costs the pharmaceutical companies money from their vaccines, etc. So we really have to. Uh, I don't know how the best way of doing it, but I would tell people the major thing they're going to run into is political problems. I mean, that's what I ran into. That's how they shut me down.
Mm-hmm. And you're still there, and thank you a lot, Professor Boydale, because yeah, it's a living testimony, and I hope that we are continue this work. I know that you're still really involved in the next trial phase for your compound. And thank you a lot, really. And I
Yeah.
hope that you enjoy this show, and that the people will understand more what is the next generation of chelations, because yeah, we need to be very careful about those techniques. So thank you a lot about this, and see you soon, Professor.
Okay. It was nice talking to you.
Okay, bye bye.
Bye.